All NEET aspirants, you are listening to podcast Botany for NEET by S. Ramakrishna. In this, the topic molecular basis of inheritance, that is genetic code. The understanding of the concept replication and transcription is easy. That is, conceptualization is easy. Why? DNA to DNA replication, it obeys the rule of complementarity. Adenine pairs with thymine, guanine with the cytosine. DNA to RNA, it also obeys the rule of complementarity. Adenine pairs with it is uracil instead of thymine. In case of translation, transfer of genetic information, that is nucleotide into polymer of, that is amino acids. So, in translation, mRNA acts as a template for polypeptide. So, nucleotide or nitrogen-based language changed to amino acid language. But neither complementarity exists between nucleotides and amino acid, nor anything theoretically drawn. There is no relation between nucleotide and particularities amino acid, in contrast to nitrogen bases. But change of nitrogen base in nucleic acid, there is a change of amino acid. Suppose AAA, it code for particular amino acid. Suppose you change it to UUU, it is going to give a particular amino acid. So change of nitrogen base leads to change of amino acid that led to the development of a concept genetic code. Next, deciphering the structure of nucleic, that is DNA, and determining the biochemical nature of the genetic material, it is a exciting aspect. And proposing the genetic code is a challenging aspect because it is a probability. So it is a challenging aspect. George Gamow, a scientist, a physicist, according to him, the opinion, there are combination of nitrogen bases code for amino acid. How we can say the combination of nitrogen bases code for amino acid? Because there are 4 types of nitrogen bases in nucleic acid, DNA or RNA. And there are 20 types of amino acids in a protein. So combination of nitrogen bases code for amino acid is right. And a bold proposition by George Gamow was 3 nucleotides code for 1 amino acid. That is a bold proposition, dare to say. How it is a bold proposition? Permutations and combinations, after assessing all, 1 nitrogen base, that means 1 nitrogen base for 1 amino acid. So there are 4 types of nitrogen bases: adenine, guanine, uracil, cytosine, UCAG. So there are 4 amino acid code for. That means it is called singlet code. So it is not sufficient. There are 20 types of amino acid. So singlet code not sufficient. Doublet code, a combination of nitrogen bases in UCAG. It generates 16 codons. The first one, singlet code, 4 to the power of 1. Second, it is 4 to the power of 2. That is 16 codons are generated. But there are 20 types of amino acid. It is also not sufficient. And triplet code, 4 to the power of 3. It generates 64 codon. So 64 codon code for 20 amino acid. So singlet code, doublet code, triplet code. So it is a bold proposition. 3 nitrogen bases or nucleotide code for amino acid. Next. And proving genetic code is a triplet code is a very daunting task. That is very, very difficult task. The first time Hargobind Khorana was instrumental in synthesizing the gene artificially by using homopolymer and copolymer. Instrumental means who pioneered the first time, who synthesized the gene artificially. Homopolymer, copolymer. So, as this polymer, the nucleic acid, the DNA or RNA, having the monomers nucleotide. In case of RNA, the monomers are ribonucleotide. In this polymer, RNA polymer, maybe homopolymer would opted also copolymer. Homopolymer means similar types of nitrogen bases. Copolymer is it is with other type of nitrogen bases. So suppose in a sequence of nucleotide there are four nucleotides. In this sequence the cytosine is there. So it is a polymer having the cytosine. In other polymer, it is having the adenine. So cytosine, adenine. So in this total polymer, what do you call it? Is the copolymer. So in a polymer of RNA, it differs in case of what? Nitrogen bases. Sugar and phosphate is the same. So in this polymer, it may be with adenine or guanine or it is the cytosine. So who selected homopolymer and copolymer. Next, Marshall Nierenberg, who studied cell-free extract, that is free cells, not intact cell, in free living cells, who studied protein synthesis by using homopolymer and copolymer and OCOHA enzyme, that is polynucleotide phosphorylase. It is equivalent to RNA polymerase. RNA polymerase requires the template to polymerize ribonucleotide, but polynucleotide phosphorylase similar to RNA polymerase, but it is template independent synthesis of RNA. That is enzymatic synthesis of RNA. So the code was deciphered to be it was a triplet code according to Marshall Nierenberg. How? In free cells, suppose he used 15 nitrogen bases, cytosine or adenine or guanine, 15 nitrogen bases. In cell, there is utilization of polynucleotide phosphorylase. It polymerizes all the nitrogen bases nucleotides into a polymer. And this RNA acts as a template for polypeptide. So 15 nitrogen bases as earlier is said. So in a polypeptide, there are 5 amino acids. So there are 5 amino acids in a polypeptide, 15 nitrogen bases in RNA. So that means the 15 nitrogen bases code for 5 amino acids. That means every 3 nitrogen bases code for 1 amino acid. So 5 into 3, 15 nitrogen bases code for 5 amino acids. The code deciphered to be it was a triplet code. So the daunting task, it was proved by Marshall Nierenberg in cell-free extract while studying protein synthesis by using homopolymer, copolymer and OCOHA enzyme. It was a triplet code. That means the code with 3 nitrogen bases, that code for 1 amino acid is a triplet code. So there are 64 codons. It is sufficient to code for 20 amino acids. These 20 amino acids are the basic amino acids, the required amino acids in a protein. I am signing off from this episode. You will see in next episode that is interesting, that is the codon dictionary.